So hi, my name's PJ. Technically it's Perry, the junior, right? That's the J, is my dad, okay? I actually teach chemistry, like she said. Genetics is actually one of the things that we didn't have a chance to go into a lot of depth when I was in grad school, but we did have to go through some of it. So I did have to do a little bit of my own research and learning on some of the topics you guys have to deal with actually, right? So let's dive right in, okay? So we're gonna start off with the basics, right? The building blocks of life. We're kind of familiar with a lot of these things, right? DNA, RNA. So DNA, right? Deoxyribonucleic acid. RNA, ribonucleic acid, building blocks for our genetic coding, okay? DNA, RNA are basically made up of what we call 4 bases, okay? And so here, we just kind of see where that double helix structure comes from for the DNA. RNA actually is a single-stranded molecule, so it doesn't actually have a helical structure. We're going to see a little bit that what it sometimes can do is that it basically kind of wraps around itself, right, to give us what looks like a double helix structure, and then just kind of goes from there. So that's kind of where the basis of a lot of this stuff comes from, okay, where our genetic coding comes from, the information, the instructions that we require for our cells to kind of procreate and proliferate. The idea, right, behind pretty much how all life started on Earth was a now somewhat defunct idea, what they used to call the primordial soup. The idea was that early on, okay, in the oceans of Earth, a lot of the different complex compounds that are required to make DNA and RNA and proteins and stuff came together, right, to go ahead and create what these what we call early amino acids. And then from there, we get the nucleic acids. Nucleic acids, right, is what's actually used to make DNA and RNA. They don't know exactly how those are made. Some of them actually speculate it might have fallen from space with meteorites. The actual compounds themselves, the chemical substances. But now we kind of figure that, it probably happened a little bit more complex than that. But the idea is that, yeah, complex life required more information. And that's one of the big things that we understand about genetics and about the genome, okay, is that it's basically just information for the cell. And so more complex life required more complex information, right? So how and where this happened, they don't know. It very likely happened across Earth as a whole. It didn't necessarily happen in any individual spot. It happened all around the entire planet in different areas. One example of how they've been able to do some experiments to prove some of this is what they call the Millerurey Experiment. There's like a little graphic up here just to kind of give you an idea how they set this up. What they did is they basically took, right, water, okay, nice clean pure water, put it with a heat source, and they gave it an artificial atmosphere in a second vessel, right, with what they called the spark of life, okay? Because the idea was that these compounds, these big amino acids and stuff that eventually make DNA, we're going to have to require some energy source. And so the idea was that the energy source was lightning. So over time, they let that run for about a good 3 weeks. And over time, right, they came back and they tested what they had, and they did see evidence and proof that they had the first level, the first building blocks, right, the first level of what we call those early amino acids. The proteins that are required to start making, right, a lot of these different kinds of structures. From there, they suspect that then you start getting what's called the nucleic acids, which is what's used to make the backbones for, right, DNA and RNA. So that's kind of where that kind of went and headed towards. The idea then was, well, DNA is a very complex molecule, okay? It couldn't have just come out of this primordial soup very quickly. More than likely what happened is that they got what's called the RNA World Hypothesis. Early on, right, life at that point wasn't as complex as life now, okay? So at that time, it didn't have to have something as complex as DNA, right, to go ahead and procreate. So it used what we call RNA, right? RNA, right, would have been what would have been the predominant information exchange chemical that a lot of complex life at that time would have done. And then we're talking about billions of years ago, okay? This happened a very, very long time ago. As more complex life evolved, right, then they started using DNA, okay? Right, billions of years later, right, evolution, we eventually get to man, okay? And so early humans, right, basically migrated out of Africa across the entire planet over the course of about 150 to 200,000 years. So you can kind of get an idea from this map. Well, we eventually spread all over the entire planet, okay? And so they now have relatively complete fossil record. There's still some gaps here and there, but they have a pretty good idea, right, how our early, what they call hominid ancestors eventually evolved to go ahead and give us modern humans. And thanks to, right, genetics, and what we call forensic anthropology, they can now go ahead and use genetics to go ahead and test and figure out exactly, right, where they went, how long they were there, what they ate, how they lived. They can do a lot of this now, and we call that forensic anthropology. To a certain extent, genealogy kind of also ties into that a little tiny bit, okay? It is possible that very, very deep genealogy can actually go ahead and give you what's called a haplogroup, which can kind of give you an idea where your ancestors mostly kind of originated from, at least the broad region, whether European, African, Asian, it can kind of give you that kind of information. Eventually, right, we did discover the structure for DNA. A lot of times people learn that DNA was discovered, the structure of DNA was discovered by Watson and Crick. How many of you actually heard that? You've all been taught that. Turns out it wasn't entirely completely true. They were the first persons to publish the discovery for the structure of DNA. But it turns out it's actually this woman, Rosalind Franklin, who determined experimentally the structure of DNA using what we call X-ray crystallography. She was the one who discovered it experimentally. Watson and Crick and her boss, basically Wilkins, they kind of took credit for that. She actually passed away 4 years before they got the Nobel Prize for this particular discovery. So it wasn't just necessarily Watson and Crick. They're the ones who attributed to it because they published the material, but it was actually this fine lady who actually determined the true structure of DNA. Thanks to that, okay, we were able to then get what's called the modern biology, what they sometimes call doctrine, because based on the structure of DNA, they were then able to understand how the molecule behaves in the cell, and then from that we get the blossoming of biotech. Biotech then led to all these companies like Ancestry.com, 23andMe, right? All that genealogical testing, forensic genealogy, so on and so forth. Thanks to her. Okay, that's kind of where it all began. Well, we eventually very quickly learned, thanks to her, that because DNA is such a very long and complex compound, that it has to exist in a very special form. And some of you may have heard this. What happens with this is that DNA is so long, if you would actually take it, right, take a single cell and take the DNA from the cell and stretch it out, okay, it would measure about 6.5 feet. Okay, that's one cell's worth of DNA. If an average human was to take all of their DNA from every one of their cells and stretch it out, it would be twice the size of the solar system. Okay, it is very, very, very long. It's also incredibly small. We're talking about the atomic scale. So the idea with that is, well, because it's so much there, that it has to find some way, the cell has to find some way to go ahead and pack in all that information, right? Because remember, all DNA basically is just information, okay? So we have what's called the chromosomal DNA, right? What happens is the individual DNA strand, right, double-stranded DNA, wraps itself around what we call histones, okay? These small little proteins, right, that kind of look like a ball. And so the DNA wraps itself around that ball, and then each individual ball basically wraps themselves around each other to then compact themselves into what we call, right, the chromosomal DNA. So this here, right, this strand, these little proteins, is what we call chromatin, and then eventually we get, right, the chromosome, okay? So I always try to, I like to use as much analogy as I can for some of this. You can think of the chromosome, right? You have strands of yarn, right? The individual DNA molecule, the double-stranded helix, DNA, yarn, intertwined into a larger thread. The thread, right, which is the chromatin, okay, then gets spooled back onto, right, that bobbin, that big spool that you have that actually holds onto all of that DNA. So, gotta have it show and tell, right? Have to have something to have some hands on. So right here is a little sample that I have of, okay, DNA in the form of, right, chromatin, right? This is DNA that is still attached to those proteins, okay? If I wanted to try to take the DNA and remove those proteins from it, the DNA would basically just completely break apart into solution. So we have these proteins, right, these chromatin proteins, these little, right, protein ball, and we have it in this solution, okay? This is actually plant DNA, okay? Because getting it from like an organism like us, a mammal or whatever, it's very kind of hard to do. This is just plant DNA, but it exists in pretty much the same way, okay? And so I'm going to go ahead and pass this around. If you want to go ahead and take a look at it. All right. Before I move on, any questions? Yeah?
Okay, are you going to go back and talk
Oh yeah, that's coming up. Yeah, that's coming up. I'm working up to it. Yes?
Hold questions about structure of chromosomes and stuff like that.
Oh, I'm going to go in like another slide or so, but then go ahead. Yeah, we can go ahead and go over those questions. Anybody else? Yeah?
Curious, so your DNA would be the same when you're born until you die, or does it change?
It doesn't necessarily change. Now, there can be mutations and stuff, like that's kind of like the, what causes cancer, things like that. But the DNA that you're born with, if nothing happens, right, would be the DNA that you pretty much die with. Oops. I don't know what I did. There we go. All right. So, DNA basically has two sets. Okay, chromosomal DNA is in two sets. What we call autosomal. Okay, autosomal DNA is pretty much all of these chromosomes that are numbered. Okay, every human being has a total of 22 chromosomes plus 2, right, that determine sex. So male DNA is set up with the 22 autosomal chromosomes and then one X and one Y. Okay, female DNA, 22 chromosomes, right, but she has two X chromosomes. So the Y chromosome is specifically what determines, right, if you have a boy or a girl. Okay, and so from that information, we basically get the basis of inheritance. Okay, and we're going to go into a bit more detail on that, but I like to show videos. Okay.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated. Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So, that whole process of where the DNA is replicated, okay, the chromosomes essentially go ahead and mix up a little bit. That's where we get our admixtures from. And that's where the actual recombination occurs, and that's how you go ahead and get, right, the basis of our inheritance. Okay? Right, this was all started by this guy, Gregor Mendel. Right, he's the initial discoverer right, of genetic inheritance, right, he used pea plants, of all things, to go ahead and prove this. He basically showed that, right, different traits from different organisms were combined and the offspring had, right, some combination of those traits, right? And so that was where he determined that genetic inheritance is actually quite complex. I'm pretty sure a lot of you probably already learned this, trying to go through genetic genealogy, right? It is quite complex. There's a lot of details on this that we're not going to have time to go over. And admittedly, some of those details, like the deeper details, I looked into, and even I'm like, okay, I don't remember that from grad school because we never got into that. But there is a lot of information here. Right, so of course we know inheritance, sexual reproduction, okay, as a result of meiosis, where the chromosomes essentially go ahead and mix up, okay. That mixing, right, is part of the reproduction process. That's where you get those traits from, okay. And of course we understand, I think a lot of us kind of heard the term recessive and dominant genes, recessive dominant traits. All right, that's what Mendel was talking about, and that's where the basis of inheritance come from. We now understand that process is driven by the chromosomes when they split up to go ahead and create more cells. When they reproduce, the chromosomes basically kind of mix together a little bit, and it's a somewhat complex, somewhat random process about exactly how they mix and where they mix, and that's why every person's an individual, okay? But because a lot of that shared genetic material is the same from mother to son, from father to son, right, then it's going to be a lot of that commonality, okay? So we typically say that the offspring of a parent is going to have at least 50% of the same genetics as their mother and father, okay? And that's kind of where we get a lot of that stuff from. And we're going to talk more about that when we talk about centimorgans, because I know that's a big topic for a lot of you, which was actually new to me. When I learned genetics, they never talked about centimorgans. We called them something else, but the concept is still the same, okay? There are 4 types of inheritance DNA. I think a lot of you probably already know this, right? 4 types. We're going to talk about those 4 types in a little bit more depth, right? We have what we call autosomal DNA, which is those 23 chromosomes that are numbered. 22 chromosomes that are numbered. Then we have Y DNA, okay, which is the Y chromosome, X DNA, X chromosome, and then we have what's called mitochondrial DNA. So mitochondrial DNA is this very special type of DNA that only comes from the cell's mitochondria. Okay, we're going to talk more about that as well. These 4 types of DNA can be used, right, for your genetic testing when you're going ahead and studying genealogy. We can kind of get an idea from this inheritance chart how a lot of this stuff works. And again, I also have these slides. Okay, Suzanne has these slides now. If you want to get a copy of them, she can go ahead and email them out to everybody if you need them. Okay. So, what do all these terms mean, right? Autosomal, right? Autosomal DNA, genetic material passed along through the non-sex chromosomes 1 through 22, right? X and Y, right? Those are the ones from the father for the Y, right? From the mother, from the X, right? During procreation by the sex hormones. Then, of course, we have mitochondrial, right? That's strictly found in the mitochondria, which is known, we typically call them the cell's power plants, and that only comes from the mother. It only comes from the mother, okay? And that's going to be pretty important for us. And then haplogroup, right? The best way that I can think of putting the haplogroup in terms of, simple terms is that if you were to take the entire human population on the planet and you were to basically plot them against this giant heritage tree, okay? Your haplogroup would be like the village, right, that your family came from, which would be equivalent to a branch off of that tree, okay? And so if you can do deep enough genetic inheritance testing, it can actually start telling you this information, right? What region, right, in Italy or Romania or Europe or Germany or wherever did your ancestors come from? The further back you try to go with this information, though, the less reliable it becomes, okay? So it can only go so far back, and that's a lot of times why once you try to get to the point of, our ancient ancestors, then you have to start going into genetic anthropology, okay, and forensic anthropology. It's no longer necessarily a genealogy thing at that point. And of course, we have our admixture, right? So this is, right, the analysis that they do, right, to determine geographical and socioethnic, right, origins based on what we call clusters. Okay, and we're going to talk a little bit about clusters in a couple seconds. Any other questions thus far? Okay? Right, centimorgans, okay? So a lot of you probably are kind of still not 100% certain exactly how these things work and what they are, correct? Right, that's at least what Suzanne was mentioning to me. In genetics, in biochem, we don't use the term centimorgan, right? It was named after after a particular scientist. We don't necessarily use that term. We use the term map unit, okay? And so what this basically means, right, a centimorgan is equal to 1 million base pairs in your DNA that you have in common between you and a given individual, okay? So the idea behind a centimorgan is it correlates to chromosomal DNA recombination mutations, right? When a person procreates, right, their chromosomes mix. That mixing is what we call a type of mutation, a harmless type of mutation. And in that course of mutating and recombination, you get new traits in that individual. Well, a lot of those traits are related, obviously, back the generations to your parents, your grandparents, and so forth. We can track these things, and they basically correlate them to what we call centimorgans. So the more centimorgans you have in common with another individual, the closer you're related to them, okay? And we're going to see a chart that I know a lot of you have looked at. And I give the example, like a parent and a child would have a large centimorgan, right, a large number of centimorgans in common, right, because they share the most recombinant DNA, right, at least 50%, okay, at least 50%. Right, so a lot of you have probably seen this graph before. There's a couple different versions of them around, but right, what this is basically talking about is, right, if I have myself on this chart, I can go ahead and map, okay, the centimorgans that I have in common with my siblings, with my cousins, my aunts, my uncles, half-siblings, half-cousins, grandparents, so on and so forth. And these have specific values to them, okay? They have very specific values. And we're going to go through, I'm going to basically talk about an example in a minute. But if you go ahead and do your genealogy testing and it says you have this individual seems to have this many centimorgans in common with you, you can go back to these types of charts and you can say, okay, if they have about, if they have 1,700 centimorgans in common with me, it's possible they can be my aunt or uncle, okay? Depending what kind of DNA we're talking about, if it's autosomal, right, Y DNA, X DNA, mtDNA. We'll talk about that more, right? If they have less, like a lot less, right, they could be like a third cousin, distant third cousin. If they have very little, they're probably not related to you at all, okay? And so if you ever have this information given to you where it says a list of people, this many centimorgans in common, and you got to say, well, I don't know how they're related to me. Like, what does that mean, how well they're related to me? You can kind of you can look up these types of graphs, and they have these average values, okay? Now, the thing about this, though, is that there's a lot of variation here, okay? Question?
Your values, do they come or coincide with plain vectors?
Yeah, that's actually where this is from. Mm-hmm.
Okay.
This is a modification of that graph that I think you sent me. This one actually is a little bit better because it shows this table where it says the average number of centimorgans in common can kind of indicate what type of relationship it is, okay? And so you can kind of map it out. The other thing is also that this graph also shows the percent of DNA you have in common with that individual, okay? This is the average percentage. And I actually like that better because centimorgan, you can have really large ranges on these numbers. And so a lot of those ranges can really overlap. And so sometimes it can become very ambiguous. Like, for example, I have this. If it's an aunt or uncle, I could have anywhere from 121 to 2,200 centimorgans in common with a potential aunt and uncle. That's a really large range. And so if I get a number, and let's say it's like, 800. Well, is it my aunt and uncle, or is it my first cousin? So I don't necessarily care for the centimorgan thing too much. My understanding is the only place that uses this term is in genealogy, is genetic genealogy. In genetics itself, they don't necessarily use this term, okay? We just end up talking about percentage that you have in common with of your DNA, okay? And so like I said, this can become very complex. There are a lot of resources out there if you need them, though. Question? Yeah.
Is this how, say you get your DNA test done and they say, oh, we've also done this other person and they've got, do they look at the centimorgans and say, this could be a relation
Yes.
to you?
Yeah. And so,
possibly in the cousin?
yes. But so the other limitation to this is that in order to do those tests, they have to have that data. And so that's another limitation that they don't necessarily want to talk about too much, or they will say it, but they don't necessarily talk about the effects of that. It's like if they only have the genetic information for a couple hundred million for like a couple million people, that's a large chunk of the population that's missing. And so again, this is where it's this is where the limitations for this can come from. Yeah.
Is that why my cousin, when I had my done, for one daughter, it says she's a first cousin to me, but her other daughter is a second cousin?
Mm-hmm. Yeah.
Because they read that centimorgan?
Yes. And again, because you have that overlap of the value, right, it can be ambiguous. Well, you know, to what degree of relation are we? Are we first cousin? Are we second cousin? Are you aunt and uncle? And that's, I think, where a lot of the other tests have to come in, okay? This is primarily based off of the autosomal tests, right? Those 22 chromosomes that you have in common with you know, no matter your sex. Whereas the Y chromosome, the Y tests and the mitochondrial tests can kind of add more to the picture to try to delineate it a little bit better about, well, who's who, okay? The X chromosome tests, those aren't very good. That's one of the things I actually knew on my own, that X chromosomal genetics is not a very reliable source for information. And so hereditary-wise, inheritance-wise, it's probably not the only thing you should go by. Right, so the autosomal test, right? So like I said, the more centimorgans you have in common with someone, right, the more related you are to them, okay? Or the higher the percentage of shared genetic traits. Okay, the higher percentage of shared genetic traits. Right, so due to genetic statistics, okay, which we're not going to get into, but there's a lot of crazy math involved in this, right? But because of that stuff, right, the specific relationships are known, right, within a certain margin, like I said, okay? Example, right, I have a cousin, right? He actually did one of these tests years ago, which is part of the reason why I eventually did one too. And he did kind of refer to me, he's like, there's this number that they gave me and it says that it's 850 and that you and I have a similar number. So I guess that just means we're related, right? He didn't know what the centimorgan thing was, but later on I realized when I was, prepping for this, I'm like, oh yeah, I remember my cousin Junior did the same thing and that's what this is talking about. So if he had a centimorgan of 850, right, from the autosomal DNA test, it statistically shows that he's very likely to be my first cousin. Right? Which is what he is. He's my first cousin. But it also shows, because of that number range, that he could also be, genetically speaking, he could be also my great uncle or my half nephew. And so it's like, well, that's not very helpful anymore, right? Unless I know for a fact how he's related to me, that's not helpful, okay? And that's the autosomal test. And so the other tests have to come in to kind of help refine that information, okay? So then, of course, you have your xDNA, right? Like I said, xDNA is not the best one. So since both parents have X chromosomal DNA, right, because your mother and your father will always give you X chromosomal DNA, a child could get a bit of both from each parent, right? If the father passes the Y chromosome, the child is male. If it's the X chromosome, the child is female, okay? Therefore, a male child gets all of his X chromosomal DNA from only his mother, okay? So that part is somewhat useful, up to a certain point. After a generation or two, an X chromosomal DNA test, it's not that useful anymore, okay? So for a generation or so, it's okay. Beyond that, though, because of the way the DNA and stuff goes ahead and recombines, yeah, then the information gets a little muddled, okay? But the good thing is male children have only matrilineal X chromosomal DNA, while a female child gets 50-50 from each parent. So if you want to use it, right, for a male child, to go ahead and determine matrilineal lineage, it can be somewhat useful for that, but only for a couple of generations. There's a better test for this, which we'll talk about in a minute, okay? Questions about that? Because I believe you were mentioning to me that this is actually a point of contention for a lot of them.
Well,
Yeah.
yeah, for a lot of reasons. I mean, there's a lot going on with DNA right now in genealogy.
Yeah, because even when I, during grad school and stuff, when I had to study a little bit of genetics, it was for simpler organisms, but the basis is the same. And when it came to the X chromosomal DNA, a lot of us, in the graduate school in the field are basically like, yeah, we're not going to worry about that, because there's just too much there. There's too much variation. there's too many factors. It's just not necessarily worth going through. I think now that the technology has gotten better and a lot of the genealogy companies have ways and tools of dealing with this, it's probably a little bit easier to work with. But again, it has that limitation. It's only good for a generation or two. Okay. Y DNA, right? That's good because, right, since only the father has a Y chromosome with DNA, only his male children will inherit that genetic material. And so therefore it's patrilineal, okay? All right? And so as a result, you can use, okay, a Y chromosomal DNA test to go back further, right, in the generations on the father's side, on the patrilineal side. So that's good for that. And so an example of this, bit of a crude example, right? If my cousin Junior and I, right, share a high, right, centimorgans of patrilineal DNA in common, right, we are therefore very likely to be related through my father's side, okay, right, or through specifically his brother, my uncle, okay, because it's patrilineal, right, because we have a common grandfather. Then, of course, the last one we talk about, mitochondrial DNA, okay. Many years ago, this was a difficult thing to do, but now, thanks to more advanced technology and genetics, right, this is becoming more and more commonplace. And it's actually a very, very powerful genetic testing that we can do, not just necessarily for genealogy, but just in biogenetics in general, right, where we have mitochondrial DNA, which only comes from the mother, okay, and is only passed down the female line, so that is matrilineal, okay. And so with this, and then also with Y chromosomal DNA, you can trace back father side, mother side, okay. With the autosomal DNA, those 3 DNA tests are pretty robust when it comes to studying inheritance and genealogy. That way the X DNA stuff, you don't have to worry about it so much, okay? You don't have to worry about that so much because it's very limited. So in this example, if Junior and I share no mitochondrial DNA, it makes it even more likely, right, that we are related on my father's side. And so taking those 3 things into account, you can eventually try to start sussing out that, okay, you're not my great uncle, right? You're not my half nephew. You are most definitely my first cousin, okay? Because those centimorgan values have such a wide range, and it can be somewhat misleading at times. You have to take all of that into a broad picture and bring it all together, okay? Questions so far? Mm-hmm.
So if you're trying, so as a female, and if you're trying to figure out maternal side, autosomal is a good place to start.
Mm-hmm.
Is there another one should take?
For maternal side?
Paternal.
For paternal side. That would be the Y chromosomal DNA.
Why?
The Y chromosomal DNA is what you'd use. Yeah, and so that's the issue then, right? If you're trying to find it for a daughter, right, on the paternal side, there is that blind spot. So at that point, you would have to try to eventually, yeah, if she has a sibling, a brother, then you can use that as kind of like a backdoor to the information. Otherwise, at that point, you'd have no choice but to use the X chromosomal DNA.
Or you can go up one or
Yeah.
2 generations and then go over and trace
Yes,
the male line.
exactly. And that's actually where I was going to go ahead and show the last slide, right? This is an example of using mitochondrial DNA, right? So because it's only from the mother, okay, we can kind of see how it goes down. And so imagine then if you're like one of these people, right? You're a great-grandson or whatever. Well, you can only see on the mother's side, but you can always go over a little bit and kind of see from the other side. So if I have a sister, okay, again, it's that same limitation. But let's say you can go over another side of the branch, and you can kind of see how that relationship goes. And it might take a little bit of doing, but, right, to overcome what we call that genetic inheritance blind spot, you can slowly start to piece together things, okay? You can piece together some of it. Like I said, this is a very complex process. I actually didn't realize completely how complex it was until I started doing my research on this. I knew it was complex, but I didn't know it was this complex, okay? Good question, though. Anybody else?
Basic questions.
Mm-hmm.
I don't think I can wait any longer.
Yeah, go ahead.
Okay.
Well, this is the time for questions, right? Because that's pretty much the end of that.
Oh, cool.
So questions, comments.
Thing that's driving me crazy is
Mm-hmm.
there's so much pairing when you talk about DNA, and then an author will just give a number. Like
Mm-hmm.
you gave us a number of 6.5 feet for the DNA. Now, is that for 46 chromosomes or 23?
All of it. That's the entire human.
46.
Yeah, it's the entire human genome out of one cell. a single cell.
Cool. Okay. During recombination, do we know if a chromosome breaks in between genes? Are genes preserved intact during recombination?
So they're supposed to be. We hope that they would be, but not always. And that's kind of where the recombination occurs. That's how you get people who have different traits from their mother and their father, right? Because now when those chromosomes basically mix in the process of procreation, when they go ahead and replicate, then sometimes the genes don't necessarily overlap perfectly when they have to be copied. And so you have a variation then in the gene between one person and another.
And you're sure about that?
And we're sure about, they're pretty sure about that,
Because,
right?
like, chromosome 1 has around 2,000 genes.
Most of them are what we call conserved, which is when they get copied perfectly to the next organism. But there's always a little bit of variation there. And so in that process is when, for example, like a person might become genetically predisposed to certain kinds of illnesses because those mistakes in an earlier generation were passed down to further generations because that little mistake must have gotten copied at some point.
And there are natural mutations.
Oh yeah, there's natural mutations as well, natural mutation cycles. Those are much slower. Yeah, those tend to be slower. Those take, hundreds of generations to occur. Yes. On my father's side, okay, I'm actually half Gypsy, right? He was a Romany Gypsy. And most of them are dark hair, dark hair, brown eyes, right? Pretty much all of them. They all look almost exactly the same. They're very Eastern European, olive complexion, right? Straight black, dark hair, right? Whereas my grandmother, on my father's side, my paternal grandmother, she had blonde hair, blue eyes. And she had a very fair complexion. And so a lot of them, they're like, what? And it turns out, well, because at some point in her previous generations, right, her ancestors lived in Russia for a pretty long time. And so there's a little bit of intermarriage at that point. And so that's what they call a recessive trait. It eventually came down where her chromosomes, right, during her, in the procreation process, the genes, the chromosomes, when they mixed, they just so happened to mix to pick up the recessive trait rather than the dominant traits. And so she had blue eyes and blonde hair. Okay, yeah.
Yeah, that's a real common thing, especially, like a couple of years ago when we first started this lab, people were just starting to do their DNA testing. One of the questions was, I don't understand, my brothers look so different than me, and our DNA, numbers are so different, And one of the analogies, and it's probably a very simplistic analogy, but one of the analogies I used to use is if you use blue marbles to represent your father and pink marbles to represent your mom, and you took all those marbles, like let's say there's 100 for your father, 100 for your mom, and you put them into a mixing bowl, mixed them all up, and then you poured them out. That would be your first child, and then you mixed them all up again, and you poured it in a second pile, and that would be your sibling.
Yes.
Now, it's the same mom, the same dad, but the arrangement of the marbles.
Exactly. Yeah. Exactly. Exactly.
Child 1 to child 2
Yep.
to child 3 to child
The amount of genetic material was still 50%. It's just the arrangement of the genes. And so that's again what Mendel basically described. He's the one who found this out by crossing the different types of pea plants, and that's where you can see those what we call Mendelian genetic tables. All right, that's kind of where that came from.
That
Yeah.
slide that had Mendel's.
Yes, exactly. Yeah.
And showing that the recessive trait shows
Uh-huh.
up in that quarter.
Yes, exactly. So there we go. And so you can see that here, right? You can imagine, right, my blonde-haired, blue-eyed grandmother versus everybody else from her generation. They all have the more common traits. She had the recessive trait, and that can happen. They all share the same genetic information, and it's a very specific known percentage of how much, but it's the arrangement of that genetic information that causes that variation, okay? I had a question over here, yeah?
It was me. It's probably stupid, but I've always heard that you can only reliably count like 5 generations back because it's so diluted when
Yes.
it gets down to you.
Mm-hmm.
So how does somebody say, I have proof that my 10th great-grandfather is related through DNA? How do they know? Because you don't have DNA for the great-grandfather, 10th great-grandfather.
That's where the genetic statistics come in, okay? That's where a lot of that math comes into play,
part of Europe or whatever that
Yes.
they came from?
Yeah, that's where the math comes in that the companies will basically do for everyone. And they know from the statistics, because of the way the DNA recombination works, because of mutation rates, all of that information, they can calculate that information within a certain degree of certainty. And that's kind of where things can get messy, because it's like, well, how reliable is this data? Because when I got my genetic testing done, it gave me a lot of that information, and I was able to look at some of the statistics, and I have a little bit of understanding of that. And I kind of did poke a little bit at it. I'm like, well, but what about this? It says like I'm 1% Polynesian. I'm like, is that statistically relevant? No, probably not. Because whoever that ancestor potentially could be, there's no genetics for that individual, right? It's just statistically probable to a 1% degree that I have an ancestor that was Polynesian. Okay, that's kind of where that comes from. And so there are limitations to this.
There's no definitive cutoff, like you can go 5 generations or 3 generations or 6.
Not from what I understand, no.
Well, there's probabilities.
Yeah, it's probabilities.
And also I wanted to point out that all the genetic testing companies will always be the first ones to tell you, don't rely on just DNA testing alone. You have to supplement that with your research,
Yep.
your books and your history and
Yep.
so on and so on and so on. You should never use DNA as a end all be all. It's just another tool in the genealogist's tool chest.
Yeah, you can think of the DNA testing and the genetic testing in terms of genealogy as like the framework for the picture that you're going to paint, right? That's the framework. You have to come in with the brush, and you have to fill in the gaps. You have to put in all those details. When I had my genetic testing done, I actually did have a couple people reach out to me asking if we were related. And thankfully, because the way they thought that we were related, I knew enough that I'm like, no, we're not cousins. We just happen to have some things in common. And it just so happens that I said, well, we could be like distant cousins, but the way you're specifically asking if we're related in that fashion, no, we're not. But so you gotta get all of your resources together to try to suss this out. Because again, this is complex and it's limited. And like we see, right, the further you go back, the harder it is to really, really use just genetic information to determine exactly how someone's related to you. It's enough to say, like when I did it, it shows that on my mother's side, right, they originated from Northern Europe. And they probably started in that region anywhere from like 400 to I think it was like 500 years ago. Okay, that deep genetic information is enough to say at least that much. But can they say specifically, well, this person 400 years ago is your great granduncle? No, they can't. Okay, they can't necessarily do that. Yeah.
One question, specifically on that, and I guess I'm delving into the forensic DNA. Can they take the wooden teeth of George Washington?
Mm-hmm.
Okay, obviously lots of saliva. Would the DNA still be good enough to
Probably not.
test?
Probably not. Not from a source like that. What they could possibly, right? Hair can the DNA in hair could potentially survive long enough depending on the environment it's in. Saliva, no, that would break down pretty quickly. Cellular material, probably not, no. Bone marrow, though, that's actually a good way they can get long-term DNA, is if the bone marrow is intact, right, they can get it and extract DNA from that, and they can do some genetic testing. But again, the older it is, the less reliable it becomes. Okay, the very much less reliable it becomes to get those deeper details. Yeah.
I read that that testing on DNA hair samples is going to come in not too distant.
Mm-hmm. I think because a lot of this genealogy testing is basically born out of forensic testing, right? They eventually realized, like, well, they do all these forensic tests and stuff. Why don't we just apply that to the ordinary population and just take it further? We have the tools, we have the technology, right? And they can go ahead and do that now. So a lot of that comes from that. So eventually, yeah, they can eventually do hair follicle testing for DNA. They can do a lot of different testing for DNA. There's other things that they can get if they really, really want to push the envelope, yeah.
So if you want to be, forward thinking, save a sample of someone's hair And make sure you get that follicle. Just get that follicle and put it in an envelope.
And put it in the freezer. Put it in the freezer. So the one thing I will leave you guys with, right? If you have any additional questions, you can go ahead and reach out to me. This is my TMCC email. But one thing I wanted to mention, I'm a big science history buff. Like, I love science history when it comes to just any field of science. And so if you want to get like a good book that just talks about, in simple terms, like you don't have to have like a doctorate or anything like that. This guy, right, one of my favorite science authors, his name is Sam Keen. Okay, he wrote a book called Violinist's Thumb. It talks a lot about the history and stuff behind, what we call molecular biology, which is the overarching field for this particular type of stuff. Genetics, genealogy, discovery of DNA structure, right, all of that. So if that's interesting to you, go ahead and check that out. It's on Amazon, right, and I'm pretty sure we might actually have a copy of it here in the library. I think I saw a copy of it floating around somewhere. Oops, sorry. Yeah, and so, yeah, there we go. Any other questions? Yeah.
Well, I have a question. I grew up in Vallejo, California, and I knew Mike Michaud, who was one of the victims of the Zodiac. Now, this GSK, Golden State Killer, was analyzed his DNA all the way back to his ancestors. That was done within a couple of years. They've had Zodiac DNA from letters, a strand of hair under one of the stamps, spit on the envelopes, etc., since the early 70s. Why haven't they caught him, and what has been done so far?
I wouldn't have any idea on that. That's a law enforcement thing. I don't know about that.
Do you know what? That's a great question for.
That is a good question, though.
That's a great question.
That is a good question, though.
Certainly, the Zodiac was every bit as important as the GSK.
I think probably the safest answer I can give you on that is there's probably a lot of other factors that are limiting them from being able to make a complete, clear, 100% case that they can actually go against someone legally. It's like they don't have, it's just like bits and pieces of information. They have to have a bigger picture, so they don't have circumstantial evidence to take that and say to a judge, hey, we want to subpoena this person because we think it's this guy, here's the proof. I think what that is they don't have enough to piece it to piece it together and directly pin someone on it. But again, it's also the limitation of they can have all that genetic information. If they don't have a database somewhere that has genetic information to match it, then they can't point a finger at someone. The Golden State Killer, if I remember right from the news articles that I read, is that someone did something, or he did something, or someone in his family did one of these genetic genealogy tests, and they were able to get from that the data that they needed to pinpoint it. The Zodiac Killer, they may not have a database that they can go and say, hey, we have this genetic profile, let's go in a database and find a match. They probably don't have a match because nobody's done it in his family. That's probably the limitation on that. Anybody else? All right.
All right, let's get
Well, thank you so much.